Sorry about that. I wasn't watching my time on that last video. So, yeah. So again, the space between the vestibule and space between is part of the glottis. Folds in the mucosa, two are attached, and so these are what actually, when air rushes past, sound is really generated into. Like I said, you're actually going to be able to, and then your lips and tongue will form the sounds that are coming, emanating from what we used to call, or larynx used to call, our voice box. All right. So again, they oscillate to produce sound. These two vocal cords. All right. So the trachea is a fairly long singular tube in which again air just continues moving down. It's got these C-shaped rings of cartilage, so we use this to demonstrate hyaline cartilage when you guys were in A&P 1. And so again, we continue with this pseudostratified columnar epithelium with lots and lots of cilia, air moving in the space, lots of mucus secreting glands here in the submucosa, and then really, instead of a muscularis, you have the hyaline cartilage for support. All right, in front of the esophagus, really has to be this way because, of course, air has a lot lower density than liquids or solids. So when you are inhaling and exhaling, and the pressure changes that occur in these tubes, you could collapse them without the cartilage for support. And the cilia here actually move in a different direction. So cilia in the upper, what we call the upper respiratory tract above the larynx, tend to move downwards, sweeping mucus back down, of course, into the esophagus. You swallow a lot of it. You produce a lot of mucus, you just swallow it. In the trachea, we can't continue to push mucus down because the tubes are going to get smaller and smaller and smaller, and you're going to block them. So it turns out that the cilia in the lower respiratory tract, that is below the larynx, will actually push upwards to try to get, again, the mucus into the esophagus so you can swallow it. All right, the bronchial tree is an extensive tree. So we look at this and we think, upside down. You've got just two major branches here because there's only a primary that goes into the right and a primary one that goes into the left. But once we get into these lungs, we're just going to branch and branch and branch. So it's like looking at an upside down tree. Or I liken it to broccoli just because of all the little tiny, tiny, tiny spheres at the ends of the broccoli. Broccoli remind me of alveoli. So we're leading from the trachea, and we're going to have to get smaller and smaller and smaller to get down to the alveoli so we can exchange. So similar theme to what had to happen with capillaries and the cardiovascular system. You start off with these big, strong tubes that withstand pressure and allow flow. And your idea, though, is to keep whittling away at the walls, getting thinner and thinner and thinner till we finally get to one simple squamous cell to make up the wall of the structures. And that would allow the most efficient exchange. So you're going to see the same theme here. Left and right lungs become independent at this point. So again, they shared air through the trachea, but once we project in, each lung is really independent and will continue to become independent with each branch. So there are 3 lobes on the right, and the 3 lobes are independent from each other, and there are 2 lobes on the left, and they are independent, and then they will continue again to branch. So primary bronchi enter at a hilum or hilum, which is an indentation where a number of different tubes enter into a structure, leads to secondary, tertiary, and lots of intermediate bronchi. When the tubes are smaller, they become bronchioles. So just like arteries led to arterioles, you get bronchi leading to bronchioles. And this cast shows some of the extensive branching a little better, but still you're not getting down to the microscopic. This has been a set of lungs that were injected with a variety of different colored latex, and the latex entered into different areas of the tubes. And just shows again how each area is really, each color is independent. So successive divisions, right, as a conducting division moves air to the respiratory division, we see many branches occurring, and so there's a difference. The right has 10 tertiary segmental and the left has 8, but none of your models in lab are really made to show the tertiaries faithfully. The cartilage is going to be maintained for a period of time, but eventually we're going to have to lose that because the cartilage is going to interfere with exchange. So As we move further into the bronchial trees, we see smooth muscle begin to predominate and cartilage go away. And this smooth muscle then can actually control flow. The cartilage wasn't really able to do that, so it's good that we can get to a point where we can, bronchiodilate and bronchioconstrict to help us with movement, controlling movement of air. We just talk about from tertiary these so within intralobular bronchioles, so lots and lots of those, generally about 22, 23 more branches. And then we get to the last part of the conducting division called the terminal bronchioles. And one of your models shows this really well. In the terminal bronchioles, there tend to be 50 to 80 of these per lobules. So again, when you go back into lab and look at the lung models, you'll see that there's these lobules on the surface that you can recognize. And the lobules tend to range from about the size of an eraser on a pencil to about a penny in diameter. And all I'm saying here is that if you scraped away the outer connective tissue that really makes the lungs look solid, you would see beneath it just about 50 of these tiny, tiny tubes that are leading directly into the respiratory division. So in the respiratory zone or division, here's what you see. So again, terminal bronchial, end of the line, and here's that smooth muscle rings. So as we come in, and this terminal bronchial branches, well then it branches into respiratory bronchioles. All right, so you see where they're showing these are respiratory bronchioles. And the respiratory bronchioles set aside because there's actually alveoli on the sides of these ones. They don't show them very well here, but that is true. Then from the respiratory bronchioles, there are about 2 or 3 more branches and they'd be called alveolar ducts. So here's our respiratory bronchiole here, and it's branching into an alveolar duct. And the alveolar duct would be completely surrounded by these alveoli, which would collectively be an alveolar sac. So not nearly as much to consider when you think about the anatomy, but the reality is most of the tissue of the respiratory system is here, by far and away in the respiratory zone. There's about 300 million alveoli between the two lungs. And this is where, again, exchange can occur. So here on the cartoon, they show you this extensive blood vessels, capillary beds that we call the pulmonary capillaries, where, again, from the right side of the heart, we would have pushed blood that was low in oxygen and high in carbon dioxide here. And in these alveoli and in this respiratory zone, we can pick up oxygen and get rid of carbon dioxide. So that's why you see this change in color as the blood then returns to the heart, to the left atrium. All right, so here's an actual, plenty of slides to show this detail. And again, lots and lots of alveoli, and alveoli back to back, and then, collectively alveolar sacs. So here again, there's some elastic in the walls. It does not interfere with exchange. That lets these stretch. They do have pores in them, so you see a couple pores here that let them connect to other alveoli. So if there's a blockage in a tube here, we don't lose the exchange here. These ones can exchange with these ones back here that are receiving from another tube. All right. The alveolar sac has type 1, which is most of the structure, simple squamous cells. Type 2, which are going to help you secrete surfactant, right? And they show you an example here. And then lots and lots of macrophages, because again, the air coming down here is coming from the outside world, so it can still have pathogens and debris that needs to be taken care of. All right, so I said again, type 1 are the majority here. They're simple squamous epithelium with some elastic in the basement membranes. This means they can expand and recoil. And this is going to be very important when we inhale and exhale. And then type 2 secretes surfactant, which will help reduce surface tension. And surface tension is a force that would actually collapse the alveoli. So if there's not enough surfactant, your alveoli will collapse when you exhale. And then macrophages protect. All right? So histological changes support this function. So again, we're reviewing this. You start as cartilaginous plates and rings, with ciliated pseudostratified columnar and lots of goblet cells from mucus. And then we're going to lose this cartilage. Smooth muscle is going to be found until we get to the alveoli. We'll see smooth muscle pretty much all the way down through the terminal bronchioles, and even some in the respiratory. And then we've just got simple squamous with elastic fibers. Cilia and mucus are replaced by macrophages because, again, the mucus would block those tiny tubes, and that would be a problem. The respiratory membrane, which consists of, a simple squamous cell in the alveolar wall, type 1, and then the basement membrane and the simple squamous cell in the pulmonary capillary wall, is only about a half a micron. Sorry, that's not a millimeter. Darn it. Micron thick, so very, very thin. And it is optimized for exchange of gases through simple diffusion. That's what we're going to use to move oxygen and carbon dioxide. So when we look at the lungs themselves, all right, and their job is, again, is to perform the exchange of gases with the atmosphere collectively. All right, so in each part has a role in this, but that's its job. So the lungs house conducting and respiratory zones, right? So coming into the lungs, the bronchi are all part of the conducting. Pleura, that's a name of the serous membranes that surround each lung, attached to the mediastinum, this area in here, and enclose. The base is on the diaphragm, so the diaphragm would sit right here, and it's totally enclosed by ribs. So just giving you a little bit of a, how does this look? The right lung, again, has 3 lobes, and the left has 2. The lobes divide into lobules, I said, and beneath the lobules would be the terminal bronchioles, but that's a smaller and smaller and smaller division. All right. When we look at the lungs, of course, we have lots of blood, and of course, we have lots of nerves to control this. So the nerves would control the diameters of the smooth muscle in the tubes, and nerves would, of course, control the diaphragm of muscle, among some other things. But the blood, again, so that we can bring oxygen in and remove carbon dioxide efficiently. So pulmonary circulation devoted to delivering low oxygen blood to the lungs. The capillaries are part of that respiratory membrane. I said they're very, very small, thin-walled structures, as you knew from previous chapters. And we get oxygen in and carbon dioxide out. So oxygenated blood then returns to the left side of the heart. The autonomic nervous system controls flow and reflexes for controlling, again, the movement of this air. Bronchoconstriction is through the parasympathetic, and dilation is through the sympathetic. So again, this makes sense because sympathetic is fight and flight, and when you are fight and flight, you need more energy. More energy means more oxygen, right? So the pleura, very, very important for the function of this lung, and you have a physio-X devoted to looking at, when the pleura are damaged to kind of demonstrate this. These are serous membranes, so 2 layers, like all serous membranes, a visceral and a parietal. So they have their own, and it's separated, so there is a left pleura here, and a right pleura here. Visceral covers the surfaces and the fissures, so the fissures, the distinctions between the lobes. The parietal connects to the thoracic wall, so it's right up against these muscles that you see here and the costals. And continues with the diaphragm mediastinum, so holds all of this together. There is a small space between them, as there always is, and that, of course, has fluid. So the fluid secreted by the serous cells and the membrane reduces friction. So as these lungs are constantly moving, right, you need to control that friction of moving parts. And it provides surface tension, which holds the lungs against the thoracic cavity, because there's fluid in here, and fluid is made of water, and water likes other water. It turns out that the visceral membrane is really sticking to the parietal membrane, which is sticking to the wall of the cavity. And that's why when you inhale and exhale, you're really controlling movement of muscles, and there isn't muscle making up the lung here. I mean, there is in the tubes, but not the lung itself. But because the lung is clinging, each lung is clinging to the muscles that make up the chest cavity, you're going to be allowed to move the lungs along with the muscle when you stimulate its movement. And all this fluid in here will keep everybody together. All right? So, very important. So the process of breathing we'll pick up on the next video. But we've covered all the basic parts. So hopefully you are starting to get the idea of how